pathways that are involved in what we would call motor things, or higher order thinking things. We talked about the prefrontal cortex, the frontal eye field, the Broca's area, and the premotor area as an association area. We just did a brush stroke over motor things on the cortex. And then I reviewed with you the missing piece. I didn't review, I showed you the missing piece. So you learned in the last test section about the areas of the cortex that control muscle contraction. And you learned the names of the nerves and where they come from. And the missing piece is, where is this information in the spinal cord? So we started the same thing with the sensory piece. Sensory is a little more sophisticated, if you will, in terms of different pathways being important. For the motor pathways, it's pyramidal. All the others we call extrapyramidal, and we do not deem them with the same level of importance that we do the pyramidal pathways. On the sensory side, it's a little more sophisticated. So, in terms of equally important pathways, the one that I taught you very early in the class, as we talked about last time, was this pathway from the Pacinian corpuscle up through the dorsal root and then all the way up through the thalamus, ultimately synapsing in the cortex. This, we said, was an example of a sensory pathway. And just like the sensory stuff, just like the motor stuff can be distributed in particular regions of the spinal cord and the funiculi, so can the sensory. So I want to look at those with you as we start class today, but also want to get some really important words out in the open here. And one of those was this pathway that I ended class with last time, and that is the medial lemniscal tract, the medial lemniscus pathway. This is, if an anatomy student is going to think about dermatomes, skin regions that are represented by sections of the spinal cord. So a C5 and C6 distribution on the lateral portion of the antecubital region here comes from the musculocutaneous and comes out of C5 and C6, where the musculocutaneous turns from the bicep and becomes superficial. That's a dermatome. Where you learn these nerves going, there are skin segments represented by those areas of the spinal cord. Now, I didn't make you memorize those, but you could come up with, you could get close from what you learned about cervical brachial, lumbar sacral plexus. If you're going to say, I have been touched, you need to think that this information is passing on this tract, the medial lemniscal tract. It is a large cluster of heavily myelinated axons that carry information up the spinal cord into the brain. And you can see these guys, this medial lemniscus tract here, is synapsing in the thalamus. So you might have a tendency to think, this must be what he was talking about when he said spinal thalamic pathway, but it is not. So I'm gonna put these in categories, in neat little categories when they don't really exist that way, just to help us remember what kind of information we're talking about. Okay, so let's start here because this is the important piece of information. Let's start down here in the spinal cord. And what I want you to see, there's your touch receptor passing through the dorsal root, no surprise there. Into the dorsal horn here, no surprise there. Headed this way. What I want you to see is the axons of this medial lemniscal tract, the touch pathways, are found in the dorsal funiculus. So the medial lemniscal tract is responsible for carrying what we have been thinking about so far, touch, And that information is actually in the dorsal funiculus. Now, it's distributed in an organized way. So let me show you. So this picture is a large transverse section, and I have another one to show you. You can see here that they're color-coded. There's, how much color you want to use. These should be white. This should be gray, and this, and this, and this should all be white. These are myelinated pathways, and the circles that are drawn on these cartoons to teach this with are intended to isolate the bundles of axons that are carrying particular kind of information. So you learned this one already. This one that's not labeled here. There's your big bundle of corticospinal axons right there in the dorsal part of the lateral funiculus. That's motor stuff. And you learned, right? You distribute these things, cervical to sacral. So you have a good handle now on what kind of information, where it is for motor for the pyramidal pathways. All right, up here in the dorsal funiculus, all this white matter, we call these areas, laterally we call them the fasciculus cuneatus. There are reasons for this. I don't care that you know them for me. The cuneatus of the spinal cord feeds the cuneatus in the brainstem. It's the name of the nucleus that carries touch information. I would like for you to couple together in your mind these words: the cuneatus, the gracilis, and the medial lemniscal tract. That will build your foundation for touch. Touch information, this specific, precise pressure touch from muscles and tendons and joints, that's where this information is. It is found in the medial lemniscal tract, which is found in the dorsal funiculus, named the cuneatus and the gracilis. Now look back at this picture one more time before I go forward. See the same words? Gracilis and cuneatus? Cuneatus and gracilis? Okay, so here's the last piece of this. So I think medial lemniscal tract here, touch information, dorsal funiculus. This information is going to the brain. Now, where? Where in the spinal cord is the specific detailed information from my body? Lower and higher, is that distributed? Well, if you saw the motor, you guessed the sensory is the same way. So I showed you the spinal thalamic one before, and here it is again. So remember, this is the one we looked at before. Cervical to sacral here, medial to lateral, that's the motor one. This is the touch area, the dorsal funiculus, specific fine pressure, touch, medial lemniscal, axon pathways to the brain. This is where this information is. And it's distributed cervical to sacral, lateral to medial this time. Okay, that helps me remember it. If it helps, if it doesn't help you, forget I said it. But this is the way it goes. In the major motor pathway, cervical to sacral, medial to lateral. In the major sensory pathways, cervical to sacral, lateral to medial. You see it? It's the other way around. Motor goes from the inside out, sensory goes from the outside in. So this area you can see here is called the gracile fasciculus. That's the fasciculus gracilis, which I just pointed out to you, all these weird anatomy ways of saying it. And you can see that's pointed over here, sacral and lumbar. And you can see the cuneatus here, pointed out here, cervical and thoracic. If we look back at this picture, I've got it labeled this way. The fasciculus cuneatus takes me to T6. So could you put numbers on that for me? So the axons that are distributed through here, would be coming from dermatomes that were feeding the cervical regions of the cord, high in the spinal cord. And you work your way cervical to sacral this way, right? And so the cuneatus, this patch or bundle that's lateral there in the dorsal funiculus, takes me down to T6. So I didn't put it here for the gracilis. Well, what would you put here if I had to write numbers in here? T7 down. So the gracilis takes me down to T6, and the cuneatus takes me down to T6, and then the gracilis takes me the rest of the way. Okay? Regional information in the spinal cord. You've got something to hang your hat on now in the lateral funiculus and something to hang your hat on now in the dorsal funiculus. And I've given you absolutely fundamental key vocabulary for these. Cuneatus, gracilis, medial lemniscus, dorsal funiculus. I know, fancy anatomy words, but these are the words that will get you home and thinking about fine touch, sensory information. That's new. Now, before, when we were talking about sensory pathways, I took you through the simplest one I could do because it's named for where it goes. Now, this one goes to the thalamus too. I got that, okay? So you might want to call this one spinal thalamic, but that's not the name of it. The specific spinal thalamic tract that we talked about before, the big one, is found out in the lateral funiculus. And most of the information for sensory that is in the lateral funiculus is not really touch. Now, I know there's overlap, and anatomists will argue with this, okay? I'm just trying to give you a structure to learn with. Most of this information out here in the spinal thalamic lateral funiculus is pain information and temperature information. So you're going to remember for me, the touch stuff is up here. It's in the dorsal funiculus. The pain and temperature stuff is out in the lateral funiculus. Now, I have one more to teach you. I'm not gonna beat you up with this anterior one because there's some different ways, and some of them cross and some of them don't, and all that anatomies, all detailed stuff. I want fundamentals: touch, pain and temperature, motor. Now you know where these things are in the spinal cord. All right. Now here's the last thing. Oh, I should show you the details, right? So this one works, and the lateral funiculus, this one works just like the motor one did. Cervical to sacral, medial to lateral. So the spinal thalamic path here, the big one for pain and temperature, is also distributed cervical to sacral, medial to lateral. Just like the motor one was, cervical to sacral, medial to lateral. So out on the sides of the cord, it's cervical to sacral, medial to lateral. On the dorsal side, on the back, it's the other way around, cervical to sacral, lateral to medial. Does the saying that help at all? Cervical to sacral, medial to lateral. Cervical to sacral, medial to lateral. So out in the lateral funiculus is the way it is, and in the dorsal it's the other way, turned around the other way. And that'll help you remember where the information is in the cord. So look, y'all, if you see lesions in the spinal cord now, you're beginning to piece together puzzles where you could actually answer questions. I see a lesion here or a lesion there, and I can predict by looking in the cord and knowing where I am in the cord what's going on. All right? Now, last one. I'm going to talk about this one some more today. Out on the edges of the lateral funiculus are axon bands that go to the cerebellum. So the edges of the lateral funiculus are axons that are going to the cerebellum. Those axons are axons that are for sophisticated control of the skeletal-muscular system. And by sophisticated, I mean this is information coming from the muscle spindles and the Golgi tendon organs that is helping me locate my body in space. See if this helps you remember it. It's a very common phenomenon for an individual with a cerebellum problem to have problems positionally locating things. So There is damage to cerebellar pathways in people with Huntington's disease. And it's also, Huntington's, we'll talk about it in physiology some more, but Huntington's also has a connector into the basal nuclei, and so it's a little more sophisticated than just saying cerebellum. But it involves the cerebellum, these pathways. When a person walks who has Huntington's, you can tell, because they have trouble predicting where to put their legs. If you put something in front of them, an object on a table, and you ask them to grab it, they'll reach out and miss it because they're not locating in space. So I want you, this will help you, fundamentals, the information that is in the lateral edges of the spinal cord is this information. It is the sensory information, these cerebellar tracts. It is this sensory information, this subconscious proprioceptor information that is telling my brain how to process where I am, where the muscles, where the joints, how you position, your balance, your body, all this kind of sophisticated, that's what I mean by sophisticated. It's subconscious. You don't think to yourself, oh, better keep my head up. This is subconscious. I don't think to myself, extend knee and place foot forward. That is a sophisticated looping network of information that passes through the cerebellum. Now, students who hear this for the first time about the cerebellum being involved in muscle contraction want to know a little more detail. Tell me how that pathway goes. So before the day is done, I'm going to mention this again about the cerebellum. I'm going to wait till I get to the midbrain to teach you the pathway. But I'm going to show you a figure that's not in your book, but it's going to help you as you think about a real simple, this goes here to here to here. And it comes here and goes there. And now this circuit of information is why I need these guys. I need to be able to coordinate specifically where I'm going, where I am, my positions, my subconscious proprioceptor. The proprioceptors are the sensory pieces that bring information to my central nervous system about my muscles and my joints. So muscle and joint sensory information, that's proprioceptor information. Okay, and it's out here in the lateral edges to the cerebellum. All right, that's all I want to do with that. Next step. Cortex, white matter. Next one on the list is basal nuclei. All right, so the basal nuclei are lots and lots of fun to talk about. You got to pick something because you could go off and, about it. I had a student a few years ago, actually, it's been more than a few years ago. He's a local pain management specialist now, physical medicine rehabilitation specialist here in town. Did his residency work at the University of Alabama, Birmingham residency program. And if I said his name, y'all would know who I'm talking about, some of you. So when he got to med school, I'd always tell the students, look, I'm about to show you just the fundamentals. I just want you to see the pieces of these basal nuclei and get something in your head about their importance. Later in your life, you will learn much. So he sent me the presentation that he got in med school. It was a 2-hour lecture on the connections to the basal nuclei. It's 160 slides. So there was all kinds of stuff in there I'd never seen before. So I'm telling you there's a lot to be known, but I want to get some fundamentals out here about the basal nuclei for you. All right, names. Name them. These are gray matter areas deep in the cerebrum. So we've been talking about all this cerebral information, sensory and motor stuff. And now, what I'm showing you here in this three-dimensional image, this is not in your book, but it's a beautiful picture to show you where these structures are these structures deep in the brain. Now, you can see labeled in the middle here the thalamus. And so that's coming next. The thalamus is where we're headed. You see how we're moving? We're moving down deep into the cerebrum, talking about structures. So right now we're going to talk about the basal nuclei, which are part of the cerebrum. All right? And they are names. They are the caudate, the putamen, and the globus pallidus. These are the three major nuclei that we consider to be basal nuclei. The caudate, the putamen, and the globus pallidus. Now this picture shows you where they are. The caudate's an interesting structure. You can see the caudate, this caudate nucleus, is a comma-shaped, an apostrophe-shaped structure within each of the cerebral hemispheres. And so you can take sections in a lot of different places and see caudate. So we got to pick one. I got to ask you a question about where the caudate nucleus is. So I'm going to show you in a transverse section how I want you to orient them. Okay, this just shows you the big picture. What else I want to say here? The caudate. You can see the putamen and its relationship to the caudate here. There are obvious connections. And then you can see the globus pallidus labeled here. Now, what I want to do, I want to get you let's get some concrete description. I'm going to cut the brain in a transverse section like this, through the basal nuclei, bless you, so that you can see where they are. All right, so here's the section. See the green section here? A transverse section through the brain. And you can see here the structures labeled. There's the caudate, and there's the putamen, and there's the globus pallidus, okay? So, and here is the lateral ventricles, which you've seen before, and there's the thalamus. So, describe for me, in anatomy words, where the basal nuclei are in a transverse section of brain tissue. Where are they? Okay, so here they are. Let's describe them. The globus pallidus is the most medial. The globus pallidus is the most medial of the basal nuclei structures. You're going to see in just a second, you can actually split it up into more than one piece. But the globus pallidus is the most medial. As I work my way lateral, the next structure is the putamen. So the putamen is lateral to the globus pallidus. Now, collectively, these two structures here, the putamen and the globus pallidus, are referred to in neuroanatomy as the lentiform nucleus. All these words have Latin meanings, but you don't have to, obviously don't need to know those for me. I want you to know where they are. The three bigs are the caudate, the putamen, and the globus pallidus. And you know now the globus pallidus is a medial piece in a transverse section. You go more lateral, you get to the putamen. Now, the caudate is not really necessary for you to know because it depends on where this transverse section is taken. So we're not going to make you memorize that, because that thing, it loops back across this way. So it will confuse you. So I'm going to be happy if you can remember the position of the lentiform nuclei. And I'd like for you to be able to describe them in relationship to where the thalamus is, because that's the next big structure, big brain structure. So in this transverse section, I'm pointing out to you putamen, globus pallidus, and thalamus. Thalamus is not part of the basal nuclei, but it is in very close proximity to those basal nuclei structures. They're very close. Okay, so that's where they are in a human brain section. Now, this was the picture we started with on the discussion of pathways. I used it to teach you the pyramidal or corticospinal pathways to the lower motor neurons. And I said, this is the big, and I went to the spinal cord and I said, here they are. While I was there, I said, look, there are some other extrapyramidal pathways. We're not going to worry about those. I want you to see them with your eyes. Later in your life, when you get introduced to extrapyramidal pathways, you'll pull this picture up and you'll go, I remember Warren had this organizational slide, and I can build this organization, then I can learn all this detail. And you can open this back up and look at it to see where these extrapyramidal things are connected. All right, but this is a useful picture here, in this area right here, The thalamus, basal nuclei, red nucleus and cerebellum, they're all connected. And it wouldn't hurt you to memorize the connections here, because I'm going to give you a little bit more detail on this. Here we're talking about basal nuclei. So can you see in this picture that there are cortical pieces of information passing? There are cortical pieces of information passing to the basal nucleus. So do neurons of the cerebral cortex talk to the basal nuclei? Yeah, they do. Does that information that passes to the basal nuclei have the potential to influence lower motor neurons? It does. So there is a motor way to influence muscle contraction from the cortex through the basal nuclei. But I'm not going to teach you that one, right? We didn't emphasize this. The more popular way of talking about the basal nuclei is the information coming from the basal nuclei to the cortex. So this is a phenomenon in neuroscience we see all over the place. When you're sending information somewhere and that information has to be interpreted, then a lot of times in neuroanatomy pathways, there's looping information. This one sends information to this one. This one gets information from here and sends that information back to the original. So there's these circles of information. And you can see that here in the basal nuclei. Yes, I can go to the muscles, but I can also take information from the basal nuclei to the cortex. So the basal nuclei both receive information and give information to the cortex. The bottom line is the basal nuclei structures influence skeletal muscle contraction. And here's how we know that. The reason we know the basal nuclei influence skeletal muscle contraction is because of the number 2 funded neurological disorder in our nation, Parkinson's disease. Parkinson's disease is a loss of sensory information in the basal nuclei, specifically from a midbrain structure called the substantia nigra. Now, like I said, your book is not going to detail this, but you guys will be interested in this. This is the pathway. This is the Parkinson's pathway. So I stuck it in here for you to teach it to you. It's going to introduce another structure for you. Remember, the cortex can influence muscle contraction through the nuclei, but what you want to know is how the basal nucleus influences the cortex. You want to know how the basal nuclei influence the cortex. So here it is. And this shows you a bit more information than you need, but here it comes. We've not got to the midbrain yet, but it'll come here in just a minute. The substantia nigra of the midbrain sends information into the basal nucleus, and that information is collected in the putamen, on the lateral edges, in the putamen, on the lateral edges of the lentiform. Now, that information is stimulatory and inhibitory, and the neurotransmitters are known. Now, this information that comes in the putamen is primarily dopaminergic, meaning the substantia nigra is using the chemical dopamine to talk to the basal nuclei. In a Parkinson's patient, these dopamine-producing neurons of the substantia nigra die. That is the explanation of Parkinson's. The death of the dopamine-producing cells of the substantia nigra. Now this pathway, I'm going to show you a picture of it, is called the nigrostriatal pathway. The nigro, see it? Substantia nigra. Striatal. When you hear the word striated, what comes to your mind? Layers. You know, striped, like skeletal and cardiac muscle, yeah? Remember the striated appearance? Look at the basal nucleus. You see where the name comes from? You see the striping? Do you see the striping of the gray matter of the basal nucleus? This is why the anatomist calls it the striated body, the corpus striatum. So the substantia nigra sends information here by way of the nigrostriatal pathway. I'm going to show you on the next picture. The putamen then sends information into the globus pallidus. Now, can you see the crisscrossing bits here? I don't care about that. The information comes into the putamen lateral. It moves over to the globus pallidus medial. From the globus pallidus, it goes to a nearby structure, which I've pointed out to you 3 times now. And that structure is the thalamus, specifically the ventral anterior and ventral lateral thalamus. Okay, y'all, the ventral anterior and lateral thalamic nuclei. I got a picture coming. I'm going to ask you this in just a minute of the thalamus. Ventral anterior and ventral lateral thalamus. These are areas that influence muscle contraction. They go into the cortical areas that are sending information downstream to the muscles. And they are, incidentally, these guys are all inhibitory. Every single one of them. So this information that actually comes into the cortex helps to slow down muscle contraction. That's why the Parkinson's person has lost this inhibition, and they have these muscle tremors. This is what your muscles want to do without the basal nuclei influencing them. Okay, so substantia nigra to the putamen, to the globus pallidus, to the thalamus, and up to the cortex. That is the Parkinson's pathway. That's the Parkinson's pathway. That is the basic structure of the Parkinson's pathway. Now here's another shot of it. This one I will use to teach you some other things in physiology. Here's the substantia nigra. This is a cluster of neurons in the midbrain. Like I said, we haven't got to midbrain yet. This is a midbrain structure. And you can see this red and blue lines are intended to be bundles of axons, tracks. So there's 2 of them here. One of them called the nigrostriatal. This one's called the motor control pathway. And now you know the basic outline of the motor control pathway, right? Substantia nigra, putamen, globus pallidus, thalamus, cortex. That's the way the Parkinson's thing works. Now, as an interesting side note, I'll pick this up in the spring. There's another dopamine pathway that comes out of the midbrain, shown on this picture, called the mesolimbic pathway. Y'all, when Parkinson's is treated with dopamine medications, the number 1 fear in side effects is schizophrenia. This pathway, and I'll tell you a couple of experimental things. This pathway right here, the mesolimbic pathway, also coming out of the midbrain, uses dopamine. But this one goes into the limbic system, which is our emotional brain. And we know from numerous publications that this particular pathway is overloaded with dopamine in individuals who have schizophrenia. So if you treat a schizophrenic patient with a drug that diminishes the amount of dopamine in the mesolimbic pathway, these people will have Parkinsonian tremors. If you treat Parkinson's with a dopamine agonist, a drug that elevates it, you will get schizophrenic episodes. Very diverse human function, related because of neuroanatomy connections in the human brain. Okay, so the nigrostriatal system. All right, next on my list, right, is the diencephalon. So we've done the cortex here, and we've talked about the deep brain basal nuclei. As we move down from the cortex toward the diencephalon, we pass a structure that you were supposed to learn in lab yesterday. We didn't quite get finished with that, probably. Our dissection took a while. This is the major connecting piece between the right and the left brain. A structure. It's in the cerebrum. I'm working my way down to the diencephalon. in the cerebrum a structure called the corpus callosum. It is the connector, the major axonal connector with its commissural, is what they're called, the commissural fibers, connecting the left and the right brain. Its most famous experimental observation was a series of experiments done by a neurosurgeon and a very famous neuropsychologist named Roger Sperry many, many years ago, in which they transected surgically the corpus callosum of individual patients who were suffering from life-threatening grand mal epileptiform seizures. As a seizure, a grand mal seizure, as I mentioned to you before, normally starts in the right temporal lobe, spreads across the brain, and they can become life-threatening. These patients agreed to have this procedure done because some of their seizures were becoming life-threatening. So the idea was to prevent the stimulus from covering the brain. It would only cover half the brain. So they had this procedure done, and Sperry decided that he would run post-operatively. The individuals, it really did have a positive impact on their lives. And post-operatively, he decided to run a series of experiments on them to test how the right and left brain of the human organism functions. And this won him a Nobel Prize. You're familiar with this. Most people are familiar with the idea of a right brain person and a left brain person. And the reason we know about this is because of the work of Sperry and the transection surgical procedures they did in the corpus callosum. So a whole series of experiments, publications, fantastic stuff. Things like this. Patient sits in an observation place. They have them do things with their right hand and their left hand, drawing and writing words. They have them do things with their visual systems because we know about the visual pathways, which I'm going to teach you. So the medial parts cross, the lateral parts stay on the same side. So if you put things in the visual field in different places, you know if it's going to this side or that side. And so they put something up there in their visual field, they put it over here, flash it into the lateral portions of their visual field for just lateral information, and ask them questions. What do you see? Describe, draw what you've seen. Just fascinating stuff. Stuff like this. If I put an image in front of you, I just picked one. I put an image in front of you of a human face that has been built out of fruit. So you know, this famous picture is it's got a banana and it's grapes and eyes, and it's like a mosaic. They've constructed this thing out of fruit, and any of us would look at it and go, yeah, they use fruit to make a face. You'd look at that. So they flash this image up into the left hemisphere because all of that visual pathway goes to the right side, and they ask the patient, "What do you see?" And the patient says, "A human face." They flash the image up into the right hemisphere, and all that information passes into the left brain. They say, "What do you see?" And the patient says, "Grapes, bananas, apples." Fascinating. From that kind of stuff we get, the left brain is the descriptive, analytical part. The right brain is the aesthetic, the art part of our brain. All that information came because of these experiments on the corpus callosum. It is the connector between the right and left brain. All right? You can see the lateral ventricle here, just inferior to the corpus callosum. There's a membrane that you will learn about as you watch those videos that separates the two, the septum pellucidum. You already know about the connections of the lateral and third ventricle. So this is choroid plexus here, shown within the third ventricle. I don't need to teach you that again. You know about those connections. Now, beneath the ventricles here, beneath the lateral ventricles in this section, you see in this sagittal view, this lateral view, this sagittal section, the colored portion of the brain we call the diencephalon, and it is made up of three pieces. The largest mass of the diencephalon is the central region, which we call the thalamus. The largest piece is the central region we call the thalamus. Now, on the videos that I asked you to watch in lab that are on the website, Mary is going to go through this, and she'll highlight each one of them for you so you can see them. And then in the lab, when you make your sagittal section, beginning of lab next week, you can do that. The thalamus is in the middle. Now, you've heard the thalamus a lot already. In fact, from what we just said about the thalamus, what kind of information do you know is passing through here? The ventral anterior and ventral lateral thalamus. That's motor information. Yeah, it's going to influence muscle contraction, okay? But what we will learn as we go through the rest of this presentation, hopefully finishing Monday, working hard, what we're going to learn is that the thalamus is in fact a sensory relay station for everything. So you saw it when I talked about spinothalamic pathways. You saw it when I showed you the medial lemniscal tract. You're going to see it with the visual pathways. You'll see it with the auditory pathways. You'll see it in a secondary sense with the smell pathways. Everything goes to the thalamus sensory. So most texts will call it accurately the sensory relay station of the human brain. The structure just on the bottom of it, I got images to show you each of these, is a structure called the hypothalamus. So this area right here is the hypothalamus. You can see that connected to the hypothalamus is the optic chiasm, where the optic nerves cross. So the hypothalamus must be a place where visual information passes through, and it is. So visual information comes into your brain from your eyes. Where does it go? It goes through the hypothalamus. Is it going to the thalamus? You bet it is. All sensory stuff's going to the thalamus. Where else would that visual information go? Young anatomy students from last class period. What is it? Where is it? Where else is that visual information going? From the optic chiasm, through the hypothalamus and the thalamus, where else is it going?
Visual association.
Where?
The visual association.
It is, yeah. It's headed back to the occipital lobe, to the primary and association areas of the visual cortex. It's headed to the occipital cortex. Anywhere else? Where is it? Saccadic eye movements. Smooth pursuit. What structure runs that? Frontal eye field is connected here. Y'all remember that picture? The superior colliculus. Okay, so it's coming up again. So it's good to think. I saw it before. I heard about it now, and I see it again. It's a good way to learn. All right, so the hypothalamus is next. And the final one here, the epithalamus, which is on the dorsal and posterior edge here, is connected to the pineal gland, or the pineal body. All right, so let's look at each of them. Here's the thalamus. Let's see now. If I had to name thalamic nuclei, I think I could look at this picture and name those thalamic nuclei based on where they're located. Do you see that? There's no need to memorize these guys. This one's called ventral anterior, and where is it? Ventral and anterior. This is called ventral lateral, and where is it? Ventral and lateral. Now, why would your teacher point out the ventral anterior and ventral lateral thalamus in this image based on what we have learned today? Why would he point that out? I'm thinking the substantia nigra projects to the putamen, that projects to the globus pallidus, that then goes through the thalamus. Where? Through these nuclei. Those are somatic motor influencers of the cortex, the ventral anterior and ventral lateral thalamus. When you hear ventral anterior and ventral lateral thalamus, the first thing you should think is, what's the problem with the muscle contraction? All right, now if you look back here on the back side, on the posterior aspect of the thalamus, you see these two little bodies that stick off of the thalamus on the back? There is no way to do a dissection like this. This is a cartoon picture drawn by using tract tracing studies and looking at the gray matter areas. These are gray matter nuclei. These are called the lateral medial geniculate bodies. Now these are important. They're going to be part of, these two will be parts of pathways that you'll have to know. So learn it right now. The lateral geniculate body is the thalamic relay for conscious vision. The lateral geniculate is the thalamic relay for vision. So if you got on board one of these guys, you're going to the occipital cortex. Visual information comes in here to the lateral geniculate, and from there it goes to the occipital cortex. The medial geniculate body is the auditory relay spot in the thalamus. Now, where in the cortex is auditory information? Visual's back here. Where's the hearing stuff? Y'all remember this? I did it with Broca's and Wernicke's and all those areas along the temporal cortex there. That's your audition area along the temporal cortex. So from the medial geniculate, I'm going to the temporal cortex. From the lateral geniculate, I'm going to the occipital cortex. From the ventral, anterior, and ventral lateral thalamus, where am I going? To the precentral gyrus. Yeah? Okay. So we don't do all of these, but you have a smattering. You have a sense about sensory stuff in the thalamus. Here's the diencephalon. Now you can see there are several structures shown here. I took a class in graduate school, a graduate course, and the course was titled The Hypothalamus, for a whole semester. So I have to teach you hypothalamus in 90 seconds. You see how ridiculous that is? That is absurd. So you got to pick things to say to students. So let's talk about general things first. The hypothalamus itself and its many nuclei are known to be the controllers of the autonomic nervous system. The parasympathetic and sympathetic nervous systems, their functions can be traced back to the hypothalamus. The hypothalamus will be the controller. The major impact on autonomics comes out of the hypothalamus. Next, on the front side, on the anterior side of the hypothalamus, is a crossing of the optic nerve fibers. The optic chiasm serves as the anatomical border on the front of the hypothalamus. It contains visual information, obviously, the optic chiasm. This is where the optic nerves cross from the medial aspects of the eye to the other side of the brain. It was knowledge here about the visual pathways that facilitated all of Sperry's Nobel Prize winning experiments. He knew this. He knew the optic chiasm crossed here at the hypothalamus and carried visual information across to the other side. As I work my way back, we see the hypothalamus is connected to the pituitary gland. The pituitary sits on the bottom of the hypothalamus. It is the interaction between the hypothalamus and the pituitary that controls hormones in the human body. You will not be able to find a hormone system in your body that is not either directly or indirectly influenced by this connection. Any discussion of the endocrine system begins here with the connection between the hypothalamus and the pituitary. And the connection is amazingly sophisticated. It looks like this. On the front side, the connection to the front of the pituitary. On the front side, the communication is by way of the vascular system. The communication between the hypothalamus and the anterior pituitary gland is vascular. The blood collected in the hypothalamus talks to the blood in the anterior pituitary. The anatomical connection of importance on the front side is vascular. Blood vessels talk between these two. The anatomical connection on the back side, on the posterior side of the gland, is axon. It's neuroanatomical. So your textbook will call the posterior pituitary gland the neurohypophysis. Hypophysis meaning under. It is the neurohypophysis, the back side. The front side is called the adeno, the front vascular control structure. There's a medial lobe too, but that's the big picture. Front and back connectors between hypothalamus and pituitary. All right? And then you can see the hypothalamus is made up of many different nuclei. They're color-coded green, purple, and blue here. But in a section of brain, they don't look like that, do they? In a section of brain, if it's a nucleus, if it's the supraoptic nucleus, if it's a suprachiasmatic nucleus, if it's the anterior hypothalamic nucleus, what does it look like? What does it look like in appearance? Is it gray matter or is it white matter? This is gray matter, clusters of neuronal cell bodies with specific functions. All right, so here's a section to show you. You can see the gray matter areas here. There's the third ventricle. Remember how I described the diencephalon the first time? The epithalamus is the posterior roof. The thalamus is the side walls. The hypothalamus is the floor. And so here we are near the floor here, and here are a bunch of hypothalamic nuclei. There's your optic tract here. There's your pituitary, anterior and posterior pituitary. There's the blood vessel connection here, showing with these droplets, and the neuroanatomical connection there. So I love this picture. It shows all these things. And then over here you can see, this is a section that I did. A published section that Wade Warren put in the literature, and it's on Google Images. Yes? It's my one claim to fame. A section, a coronal section with my little blue arrows out of the article that I published in the Journal of Pineal Research many, many years ago. It made it into Google. I have arrived. The suprachiasmatic nucleus from a long Evans rat. So there's the third ventricle right there. And you can see it ain't that clear, is it? These beautiful little circular places showing these gray matter areas, not that clear. This picture was specifically done, this histoimage was done, to highlight this nucleus right here, a nucleus called the suprachiasmatic nucleus. And y'all know why it's important? Well, because it's important to me, right? That's why it's important to you? No, it has other reasons. It's important because this is now recognized in the literature as the biological clock. Now, I'm going to build one more connection for you. Do you guys know who won the Nobel Prize in Medicine and Physiology this year? Nobody knows. You didn't even know they were awarded, did you? You're not paying attention. Michael Rosbash and Jeff Hall won the Nobel Prize this year. Now, when I was a young man, I worked in a laboratory at Texas A&M. And Vincent Casson was my major advisor. And my number 2 guy on my committee, his name he's still at A&M, Paul Harden. Paul Harden was a Drosophila expert, fruit fly, and he worked on biological rhythms. And his lab, the Jeff Hall Mike Rosbash lab, where he worked at Brandeis University, discovered some proteins that were produced in Drosophila that were known to modify their ability to tell time. And that discovery propelled the work in circadian rhythms and biological clocks that would continue for the last 20 years in the sciences, with dozens and dozens of publications that would ultimately result in the awarding of a Nobel Prize this year to those 2 guys. Now, it is true that your teacher is some little bumpkin nobody at a little school in Louisiana. That's true. And he understands that. But let me tell you something. I could get on my cell phone right now and I could call Benny Casson. And I could get the cell phone number of Mike Rosbash. And we could get the Nobel Prize winner this year in medicine physiology on the phone in this room if we had to. Does that make you feel important? Not really. Okay. I just want to let you know, you are You are only 3 steps away from greatness by being here today. Does that make you feel important? Okay, so this was some of the original work done in Vinnie Casson's lab. This is an important structure because it runs biological rhythms. It is the human biological clock. All right, I want to say some others here before we leave this, and then we'll pick up in the midbrain next time. Other nuclei here that are important in the hypothalamus that do things, a couple of them. So there's the suprachiasmatic nucleus right there. Wonder why we call it suprachiasmatic. Why would you name it suprachiasmatic? Supra means greater than or on top of. Y'all see it? Where is the suprachiasmatic nucleus? It is on top of the optic chiasm. That's its name. Chiasm means crossing where the optic nerves cross. What about this one? This one's called the supraoptic nucleus. So it must be a little bit further posterior. So these supraoptic nuclei here, you can see them out here. Why would I call it supraoptic? You see where it is? Okay, now they're cool. In the first publication that I sent off for review, Evelyn Satinoff at the University of Tennessee wrote back and said, I think there are clock cells in the supraoptic nucleus. You're going to have to prove this to me, Wade, before we publish it. So I had to do, that's part of why this section showed up. The reason that work had to be done, because the lesions that we did had to prove that we did not damage the supraoptic nucleus. So her lab, she and Rebecca Prosser were publishing work that suggested the supraoptic nucleus also had the ability to tell time. Because the rhythm that is most hardwired in the human body is the body temperature rhythm. And we know that the nuclei, the supraoptic nucleus, are the ones that cause the body temperature to be what it is. Why do you sweat? Why do you shiver? Where did all the information start? Where does all that stuff begin? Right here, in the supraoptic nucleus. These are the body temperature regulators. So they're very important areas of the hypothalamus. Clock stuff, and here the supraoptic nucleus, body temperature regulation. Okay, one more, and then I'll shut up. I'm 2 minutes over. The mammillary bodies are considered to be the posterior border of the hypothalamus. They're also considered hypothalamic nuclei. And these guys they actually relay olfactory information. Now just tag that one because I'm going to come back to that. Olfactory information passes through nuclei of the hypothalamus. That's going to be important. All right, off to the midbrain on Monday. And I'll try to get this wrapped up Monday or maybe early in class on Wednesday. We've got to get done with the nervous system soon. You did? I think so. I don't think anybody's coming for advising at this time, so we'll try to call them. All right.